be the time when you'd be sitting in the atrium area, having your lunch while we're having the speakers give their brief presentations. So thank you for sticking through this particular session while we're all here. So what I would like to do is go over each of the speakers that you will hear this afternoon and give you a little bit of background information on why they got the NNDC Recognition Award. If you're interested in more information on their bio sketches, please feel free to refer to the program booklet. So our first awardee is from 2018. She was unable to attend the meeting at that time, and she deferred to 2020. Everyone knows with COVID, we didn't have a meeting in 2020, but she graciously allowed us to give her presentation here at 2022. So let me start with Dr. Catherine Champagne. She is Professor and Chief of Nutrition Epidemiology, Dietary Assessment, and Nutrition Counseling at the Pennington Institute. She's a native Louisianan, and she is a fellow of the Academy of Nutrition Dietetics, the Obesity Society, and the American Heart Association. She's involved in studies that include dietary counseling and dietary intake assessment, and she has interests in women and children's health, children's nutrition, diet for weight loss and chronic disease, as well as the Mediterranean diet approaches, physical activity promotion, and nutrition assessment for diverse populations. She was also involved in the final design of the DASH diet, DASH sodium diets, and she's been involved in several NIH trials, such as the Diabetes Prevention Program, the Look Ahead trial for diabetes management, the prenatal trial, the weight loss maintenance trial, pounds lost, and many others. More notably, Kathy is also a member of the NNDC steering committee. So thank you for your service. Kathy, we're so proud of you. We would like for you to go ahead and give your remarks at this time.
Thank you. I am trying to share my screen. Oops. Okay. And there is my screen, and I'm assuming that you can all see it.
Yes, we can.
All right, well, so I've entitled this presentation, Dashing Through a Career in Research. And of course, you know that because we so often dash through airports, I thought this was a good figure to show that, you know, it kind of looks like me, but I wouldn't say I just dash quite that much. But anyway, so this talk was supposed to be given in 2018 in Minneapolis. I actually want to tell you, and probably most of you know, that I had an optic nerve problem, and there was some suspicion that it might be very significant. And it took the whole month of July in 2018 to figure out that I probably had a viral infection, and everything returned to normal. So when we had the 2020 meeting, you know, we could do an acceptance, and, you know, that was basically 5 minutes. But in actuality, I was told to prepare for a 45-minute talk in 2018. And, you know, so I've cut this down so that quite a bit, so that you can appreciate what I was going to say somewhat in 2018. And this is really kind of my story, how I got interested in research, the person who got me interested in nutritional databases, my personal research journey, you know, research questions, you know, how did I get where I am now, and basically funding challenges along the way. So I've always been more interested in research than any other aspect of dietetics. Got my master's in 1972. You can see I've done a lot of different things. I guess the one thing that is kind of important is that in 1978, I did coursework at LSU to complete my PhD from Mississippi State, and thereby met my mentor, Margaret Moore. And, you know, for those of you at NNDC, she was always involved in NNDC. I worked in the agricultural chemistry laboratory at LSU. That's kind of key because I saw the opening of the building and opening of the Pennington Center. And by 1989, I was either having to give up my dietetics career or find a way, you know, to stay in the lab at LSU in the ag chemistry lab. So on December 15th, I joined in 1989. I had joined the Pennington Center. And so now I'm in my 33rd year, so it's a historical. Margaret Carrington Moore was my mentor. She actually developed a Bogalusa heart study nutrient database, extended table of nutrient values. And she retired from the Department of Health in 1964 and really manhandled that database from her New Orleans French Quarter apartment, which now is just not what it was back then. It's a little shop right now. She knew she couldn't work forever, so she was looking for someone. And one Saturday in July 1989, I called her and said, you know, got anything going on? I would like to kind of look at a career change. And really, this is important. She told people, if I had died before I found a successor, the database would be lost. But now I'm confident that it will live on. Kathy has both the chemistry and nutrition backgrounds that I was looking for. Nutrition came from chemistry, like a twig comes from a tree. And you need to know food values and need to have chemistry knowledge. So as I look back, you know, it was sort of fortuitous that I was at the LSU Agricultural Chemistry Laboratory. These are some pictures from the 1993 Data Bank Conference and other, these at the bottom, these two are from the Data Bank Conference. Some of you will recognize John Clinton, who we haven't seen in quite a while, but these are other pictures of Margaret and me when I was quite a bit younger. We hosted 4 conferences by Pennington. The first 3 shown here, the 18th, 21st, and 26th were hosted at the Pennington Center. And the 33rd National Nutrient Data Bank Conference was a one-day conference in New Orleans, which was a satellite to experimental biology, I believe. So my research career has come from a lot of places. Basically, I'm going to glance over this, but the Department of Defense really was the main source of my funding over the 32 plus years at Pennington. And, you know, I worked on Delta, I worked on DASH, and I think the really interesting thing about working on DASH was that we had to decide what database we were going to use, and there were 4. And then actually, George Bray said to me, I won the cook-off because I got the menu development for both DASH and DASH sodium, because our database was ranked the most accurate, but the databases were not statistically different. And there is a reference for that. I think it's a little bit more than the database. It's also the person who is doing the data analysis. Life After DASH really involved the premier multi-center clinical trial where we taught people how to follow the DASH diet. And we did it was an 18-month trial with biweekly sessions and monthly sessions, weekly, biweekly, and then monthly. And my job was to be the intervention director for the Pennington site. Then we were funded for the weight loss maintenance trial, which was the trial that followed. And again, the most important kudo and the one I'm most proud of being a part of is DASH. And according to the 2022 US News and World Report, DASH is ranked number 2 in the top of many things. It is not a weight loss diet. So basically, but we rank number 10 out of 40 diets. There are other lifestyles projects I've been lucky to have been involved with in my career at Pennington. MedStep, which is a comparison of Mediterranean versus the Step 1 diet for cardiovascular outcomes. That was a 6-month study. REACH was another study, reversal of early atherosclerotic changes by diet, funded by the National Dairy Council, where we looked at different types of diets with fat and saturated fat. Of course, the diabetes prevention program and the Look Ahead trial. And of course, many of you would be familiar with the Pounds Lost diet, which was funded by NHLBI. It had 4 different arms varying in 2 different types of fats versus 2 different levels of fat and 2 levels of protein. And really the thing that the outcome was that calories were more important than any other particular macronutrient in those trials. This is just a clearer picture of pounds lost and what happened. And you'll see that everybody lost weight and everybody gained weight. And this is sort of typically what we see with some of these trials. I've been involved, as I said, initially with a lot of Army projects. And, you know, I've collaborated with the USAREUM folks, the U.S. Army Research Institute of Environmental Medicine. I've been on a lot of military bases. We've done field research with MREs. We compare different types of rations. We use a visual estimation process to actually look at foods and try to figure out what the soldier was eating. And then we've really advanced to some food photography options. I have been in more than 20 studies, either field or garrison. I've listed some of them just to give you a feel for the fact that, you know, we looked at Fort Jackson, South Carolina, looking at female soldiers. We did, one of our big projects here was a menu modification consumption study, which was conducted in Louisiana at Fort Polk. Other studies, I think this one here, number 6, Great Inagua Island in the Bahamas, and I have a little bit more to tell you about that. And then some other studies where we've worked with Navy women, we've worked with Army, Navy, and we actually worked with females during recruit training. But in September 2001, we all know what happens. 9/11. So our studies were then put on hold, and then we did other studies after 9/11. So you see, after 9/11, it wasn't until 2009 that we actually did more studies. And I think this was, and right now, and I know that someone who is on this, this conference has been involved in Go for Green, and there's a poster on Go for Green. So I think that, you know, it's nice to have people that I've worked with in the past on this this webinar, this thing. Anyway, we had an Optimum omega-3 trial, which was a study in which I was the principal investigator, in which participants were randomized into one of 3 experimental diets for 10 weeks in a controlled feeding paradigm at the Pennington. We used DOD, a regular DOD diet, and then 2 that were enhanced with omega-3 and moderate omega-3 and low omega-6 foods. well, I should have got rid of my animation, but here's some of the foods that that swapped. And so we use special chickens, chicken meat, chicken sausage, different altered spreads and sauces. And of course, we had this smoothie, which was procured from Sweden. And that was one of the additional aspect of the high omega-3. We found that swapping ingredients over a 10-week period resulted in a significant change in blood levels. Arachidonic acid was decreased, and plasma endocannabinoids were also lowered. And we demonstrated actually that blood levels can be changed through specific swapping, which I have a period in swapping, sorry about that, of selected ingredients. But research is fun. So basically, sometimes we work in unique circumstances, and this provides a really memorable experience. So I mentioned back a few slides ago that I worked with the military in the Bahamas. So when I tell people that I went to the Bahamas, they thought I was on a tropical vacation. The first night was spent at a very nice hotel in Nassau. And then we went to Great Anagua Island, which has one town, Matthew Town. There was one of the people in that study that she was a challenge. And I said, well, what are we going to do about this person? And one of the people in charge said, we're going to send her to little Anagua Island where there's nothing. But you can see this was a pretty remote, this was right here at the bottom was the airport. So it was a lot of fun and a lot of, I mean, that's me, looking pretty ragged. And with my tropical stuff that was interesting. There are some other pictures I could show you from that, which would be very impressive. But anyway, how I got interested in research. I love dietetics, but I hated clinical. They were mainly because they were about to discharge the diet. And I'm walking in there trying to do a diet consult. Teaching was fun, but the students kept falling asleep in class. So I wasn't too happy with that. So research, as I learned, is always various. There's lots of diversity. There's stuff to do. And I think what's kept me motivated, as you could see from the previous slides that I've shown, is that there have been opportunities to get involved in a lot of research projects. And my experience in diet design and dietary assessment and interventions is useful to many of the people in the projects that I've worked on. So the other question is, how do I formulate my research questions? Well, because I'm in a research track, sometimes the research questions are already formulated and I'm the person who's got to carry them out. And, so if you look at steps along the research path that might be interested to someone who questions how to get into the research profession, I would say know what you're interested in and what skills you have to contribute. Find out who is doing the type of research that you're most interested in and get involved with that person. And as they say, the rest will come. So again, challenges to researchers, and we all are very much aware of this. Good ideas require funding. We have seen the shift from plenty of federal funds to real competition. State support is sometimes problematic, and we have to seek out of the box options. And finally, sometimes it's being at the right place at the right time, so often an accident as opposed to a well-drafted plan. That's my research career. I capitalized on my skills and talents, and it's paid off. And the bottom line is you need to be pleased with what you've done and what you're doing, whether or not you receive praise or awards, such as the award that I received from you in 2018. And having said that, I'm truly proud and humbled, and it's been a pleasure to speak with you today on this. So here's, I like cartoons, so basically you can look at these later since they will be posted on the recording. And with that, I thank you for listening.
Please join me in thanking Dr. Champagne for an amazing presentation today. So,
Thank you.
oh, you're so welcome. Our next awardee is actually the 2020 awardee, and that is Mr. David Hartwitz, who actually retired from the USDA 3 years ago, where he spent his entire career as a nutrition in the Nutrient Data Laboratory. David is known for having worked on several of the USDA's nutrient data bank systems and the development of Food Data Central. He has also worked on dissemination of the USDA's food composition data, as well as compiling nutrient values for vegetables, legumes, spices, herbs, nuts, and seeds. And he's even worked on some of the special interest databases that if you're working in bioactive food components, you may have actually used some of the data that he has contributed to. David is also a regional director, regional data center overseer for InFoods, and he is the immediate past chair of the Nutrient Databank Conference. So I would like to welcome Mr. David Haytowitz to give his presentation. David?
Thank you. I don't have any slides, but if you want to see me, you have to give me permission to start my camera. Okay, I will do so. There we go. Again, I would like to thank the executive committee of the conference for honoring me with the 2020 Recognition Award. In 2020, I gave a brief presentation, and the thinking was that I would give a full talk at this conference in Ottawa. However, the coronavirus is not done with us, and we are again virtual this year. A little history of my involvement. While a food science major at the University of Massachusetts in 1975, I was taking a course in food analysis. Professor held up a copy of Agriculture Handbook number 8 and passed it around the class. I thought this was an interesting book, so I copied down the information on the cover and ordered a copy, which cost $3.60. Little did I know that this book, its later versions, the database behind it, would be my career for the next 40 plus years. In the spring of my senior year, I filled out the SF-171, the US government employment application, thinking maybe I'd get a job at FDA and mailed it in. There were no online versions back then. In October 1977, I got a letter followed by a phone call from Ruth Matthews asking me if I was interested in a job at the Nutrient Data Research Branch, then in Hyattsville, Maryland. Since I had just lost my job in a QC, working in QC, a small family-owned cheese company, I accepted a move to Maryland. That was a good thing because the cheese company burned down from a fire of suspicious origin within the next year. Several months after I started work, I was told there was a conference I needed to attend in Arlington, Virginia. This was the 3rd annual meeting of the National Nutrient Data Bank Conference, which is the first one I attended. As travel funds were limited, I did not attend another conference until the 13th in Framingham, Massachusetts. I then attended the conference every year except for the 34th, which in Grand Forks, which I missed because I was going on vacation. I still not gone to North Dakota, which is only one of 6 states I've not visited. But at that time, I thought this was a way, maybe I could get data on my assigned food groups. It turned out for the most part, these people did not generate data, but were interested in what USDA could provide. The different way people use the data. You know, thinking at Gary Beach's talk this morning, I noticed here is a copy of SR3 on magnetic tape. This big reel of tape holds about 40 megabytes of data. Then we disseminated it on 3.5-inch floppies, which I think this is about maybe 1.2 megs, and there were a whole bunch of them. Eventually, we got to CDs, which hold somewhat more data. At least we could get it all on one disk, not dozens. Now, of course, it's all available on the computer and through the internet. But after working for over 40 years in the field of food composition, I'd like to leave a little what I've learned and give you a few things to consider. One, you must have good food descriptions with all the detail possible. You can have the best sampling plan, top-notch analytical methodology, but if you don't describe the food item properly, the data is useless. When you aggregate data, of course, some of the data is not pertinent to the aggregated food, but it should remain available through a drill down process to the individual data. Sometimes looking through the branded food products database, I noticed there's about 600 food items in the food description of cereal. So more precise information may be included in other fields. One should not have to hunt for that to find a good food description. Also, when grouping data from different sources, make sure you're aggregating the same food. And working on a project for FAO earlier this year, I saw somebody had merged lemon sole into the citrus fruit group. Lemon sole is a fish, not a citrus fruit. But going back to individual data for individual food items, be careful. You may have bought a lot of tomatoes that's purchased at the Shaw supermarket in Sharon, Massachusetts. So you may say, I shop at that store, I'll use that data. Wrong. The lot was purchased at least several months ago, possibly longer, before the samples were processed and analyzed. The data were then reviewed and ultimately makes its way into the database. As Dr. Hunley discussed yesterday, there are a lot of factors which affect the variability of foods. The sample you bought today, though from the same store, was probably not grown in the same location. It was grown at a different time of year, could be a different cultivar, a different fertilizer regimen, experience different climates, etc. As Gary said, as Jim Hunley said, the food you analyze is not the food you eat. Therefore, when analyzing a diet for an individual, unless you're analyzing the actual food they ate, it's best to use the mean value based on a large number of samples. And of course, the more samples, the better. Avoiding composites whenever possible. And the same is also true for evaluating data for large populations. Make sure your nutrient values make sense. Looking at the branded food products database, I see a record for a mayonnaise containing 2,160 grams of fat per 100 grams. Also, I see a sodium value of 1,570,000 milligrams per 100 grams for green olives. I know Kyle McKillop addressed some of these issues, but it seems with a database containing millions of nutrient values, some questionable values can sneak through. Hearing Dr. Jenkins' keynote presentation yesterday morning, I recall when I started USDA, scientists were arguing over the definitions of fiber and the best methods to analyze it. When I retired, they're still discussing it. Early methods, at first we were analyzing crude fiber, which actually only measured some of the fiber constituents. The latest method developed by McLaren Group measures more of these constituents using a rather complex set of extractions and filtrations to quantify a bunch of fractions and then summing their amounts to derive a value for total dietary fiber. We also get some subcategories like soluble and insoluble fiber. But each of these fractions have its own unique impact on the microbiome and human health. As a result, different foods will then have varying impacts on human health based on the relative concentration of each of these constituents. In fact, it may be advantageous to drop values of total dietary fibers and the major subfractions such as insoluble in favor of comprehensive data on the individual fiber fractions. Another issue is the use of conversion factors for protein. These factors were developed along with the Keldon method for protein well over 100 years ago. But the method actually measures the nitrogen content of food, and based on the assumption that protein is 16% nitrogen. So nitrogen to protein conversion factor of 6.25 was set. Of course, not all proteins are the same based on the specific mix of amino acids in the food. In 1931, Jones developed specific factors for additional foods and food groups. However, some of these food groups are quite broad, and if a specific factor was not available, the general factor of 6.25 was used. Also, this non-protein nitrogen was not non-protein nitrogen was not considered. And due to the limitation of the system, again, it may be advantageous to determine protein by summing the individual amino acids as FAO had recommended in 2003. Similar issues also were found when dealing with the conversion factors for calories. Again, they're rather old and they're rather broad categories. So in conclusion, there are many areas for future research and database improvement. And again, I would like to thank you, the executive committee, again for this high award. And again, like Kathy, I am humbled and honored. And hopefully we'll be able to meet in person in Ottawa in 2024. Thank you.
Please join me in thanking David for an amazing presentation. I sent messages to both of you, thanking you for your extensive work and for your service to NNDC as well. For those who may not have noticed this, we've actually heard from 3 award recipients. The 2 that you heard today, and also from Dr. Gary Beecher, who was the recipient of the 25th conference meeting in 2001. So thank you so much. I am happy to report our next session is not going to start until 12:35. So for those of you who have not had an opportunity to enjoy a little bit of lunch, you will be able to do so in the next few minutes. So with that, we will go ahead and take a brief break until the next session starts, which is session 5, expanding food composition beyond nutrients, that will be moderated by Judith Spungen from the Food and Drug Administration in the US. So enjoy your health break. We'll see you at 1:35. Thank you.